Hello students, today's session is very interesting because we are going to discuss about various experiments based on the physiological process that we have discussed in the last class. The experiment about diffusion, about osmosis, then osmosis and diffusion, then they are in the transportation of substance in the concentration gradient region, that is higher concentration to lower concentration. But today we will discuss about the experiments. As you can see that I have made an experimental setup to show, and based on this experimental setup we will discuss all the four possible ways in which we can make the changes in this experimental setup to prove that osmosis is always a process through a semi-permeable membrane, and there are some factors which inhibit the process further, because in the diffusion continues till the substance gets intermixed with the area available to it. So that in diffusion process is different from osmosis, as a semi-permeable membrane is present there, and secondly some other factors are inhibiting that movement. So in the latter part we will discuss about the tonicity of the substance. Tonicity is the concentration of the solution or the cell sap and the different regions of concentration. So let's start with the experiment. As you can see in the diagram there, this is the strand to which a thistle funnel, inverted thistle funnel, is attached with the help of a clamp, and in this thistle funnel we have taken a concentrated solution of sugar. Concentration of solution of sugar in this inverted thistle funnel, and this thistle funnel is inserted in a beaker containing water. Now, the level of sugar solution is at this point the initial level, and at the lower part of this thistle funnel it is covered with a semi-permeable membrane. Now, what are the observation? After some time, what we will observe in this experimental setup, that there is a rise in the level of sugar solution. This is the final level. So the first is the observation, rise in level of sugar. Number two, when we taste the water in the beaker, there is no change in taste. No change in taste. It means that the sugar solution is not coming out of the thistle funnel into the beaker. It means the movement is always from beaker to thistle funnel, this way. So why the movement is always from this to this? Because the concentration of sugar or the solute concentration is higher, but the solvent concentration is more in beaker. So, and osmosis is always about the movement of solvent. Movement of solvent is from higher to lower. Lower concentration, that is high to low. So this process continues for a few hours. After that, this process stops. We can see that there is no further rise in the level of sugar. So why there is no further rise? What is the reason behind that? All we will discuss. So first of all, the rise in the sugar solution is just because of the osmosis process. Osmosis is movement of molecules from higher concentration to lower, and which gives the rise in the level of sugar in the funnel, and no change in taste is because of that the movement is always from beaker to thistle funnel. Next is why the rise in level stops. That rise in level stops because this rise in column increases the weight of the solution, and this increase in weight acts as a wall pressure on this membrane. So that wall pressure, the pressure exerted by the fluid, is the osmotic pressure. Osmotic pressure and the pressure of the column is just like wall pressure, at which nullifies the osmotic pressure is nullified by weight of the column. So there is no further rise in the level. Weight of the column nullifies the osmotic pressure. It stops the further movement of water from beaker to the funnel. So that is the reason this is very important for examination point of view. Next is in the second experiment, what we are going to do is, we have changed the membrane with a rubber sheet. Now what happens? When the membrane, the semi-permeable membrane is changed with the rubber sheet, there is no net flow of molecules because the rubber sheet is impermeable to the substances. Impermeability does not allow substance to move in. So there is no rise in the level of sugar or no change in the level of beaker. Because in the first experiment, this level would go down. As the water moves up, the level in the water also goes down. So this, in rubber sheet, there is no change. The experimental setup would remain as it is. In the third, we have taken muslin cloth. Instead of that membrane, we have taken muslin cloth at the mouth of the funnel, and muslin cloth is having large pores, so that pores allow all the substances to enter or leave. All the substances could enter or leave the funnel. So even the solute particles, the larger solute particles can leave the funnel. So after some time, the level would rise. The solution in the beaker would be slightly sweet in nature. So that it means there is bidirectional movement of the solute as well as the solvent. Next is, in the next experiment, we have taken, in place of the funnel, we have taken animal bladder. Animal bladder is just like a semi-permeable membrane. So all the observation would be same as in first case. Animal bladder is a semi-permeable membrane. So the observations are similar to the first experiment. Rise in level, then no change in taste in the beaker, then increase in the weight of the column. Number 4, level of water goes down. These all points. Then one more term is related with this, is the osmotic pressure. In the next experimental setup, or just to prove that what is osmotic pressure, we have kept a slight weight in this area. Putting a slight weight on this area, what happens that the osmosis process is stopped. Why the osmosis process is stopped? Because that pressure, the weight of the substance on the funnel stops the process of osmosis. So that weight or the pressure exerted by the weight is equal to the osmotic pressure or the pressure exerted by the fluid to nullify the osmotic pressure. So in other terms, we can also say that pressure exerted by the movement of fluid on the membranous wall. So that is the osmotic pressure, and in this experiment, by the weight, putting some weight on in the column already in the starting of the experiment stops the process of osmosis. Next we are coming to the topic tonicity, isotonic, hypotonic and hypertonic. These terms you might have heard in junior classes. Let's start with the middle one. In a beaker we have taken a cell or some raisins or resin act as a cell. Inside the resin, the sugar concentration is very high, and outside this we have taken water solution, water, simple plain water. If the concentration inside the sap or the beaker is equal, when the concentration in both is equal, concentration of fluid or the solvent is equal in both, and then it is isotonic. Hypotonic, if the concentration of sap or the solvent is low inside, then it is a comparative study. Comparison between the concentration of solvent in the beaker inside the cell. When the concentration is equal in both, then it is isotonic. If it is low in cell and high in, we are discussing about concentration of solvent. So solvent or concentration of, if in terms of solute concentration, if solute concentration is high, it would result in endosmosis. Solute concentration high, it would result in endosmosis, and the solution is a hypotonic solution. Hypotonic solution, less concentrated than the solution in the cell sap. Then next is, if the solute concentration is more in this, the next one, is low. Low in the cell, solute concentration is low in the cell sap, high in the solution, then exosmosis takes place. Exosmosis results in flaccidity, or here we can write the turgidity. Endosmosis is responsible for the turgidity, while in exosmosis it is flaccidity. Exosmosis, and the solution is hypertonic. When in comparative, when the concentrations are equal in both, then it is isotonic solution. When the concentration of solute is high in cell, less in beaker, then it is hypotonic solution. When concentration of solute is low in cell and more in solution, that is hypertonic solution. This is hypertonic solution. In terms of solute concentration, movement of solvent would be from low solute concentration to high solute concentration, from low to high solute, but the solvent concentration just here, solute concentration low, solvent concentration high. High to low, this one. This is all about tonicity or isotonic, hypotonic and hypertonic solution. One more term is plasmolysis and DEPLASMOLYSIS. Plasmolysis is a condition when the cell becomes flaccid. When the concentration of solute is more in this solution, and more concentrated solution results in movement of substance solvent from inside to outside. This movement of solution from inside to outside results in flaccid condition or shrinkage. This shrinkage is termed as plasmolysis, which would result in bursting of cell membranes also. So, and when this flaccid cell or shrinked cell is again kept in a solution, hypotonic solution, the cell, if the cell membranes are not ruptured, then or not, the cell is not dead. If they are kept again in the hypotonic solution, the cell swells up, becomes turgid. That condition is termed as DEPLASMOLYSIS. So here shrinkage, again turgid. So this is all about flaccidity, turgidity, tonicity of the solution, then the experimental setup. Thank you for today. In the next class, we will discuss about other experiments to show the root pressure or the turgor pressure. Thank you.